We just finished looking at this diagram right here, worldwide distribution of HIV infections. And as we pointed out, Africa is still the hotspot. But keep in mind that this is really a worldwide problem, and the numbers are staggering. 35 million people are infected with HIV worldwide. So this is definitely a problem that requires attention. And compare it to 6 million reported cases of coronavirus. We're getting there though, especially since coronavirus is heavily underreported. But HIV presents its own problems because vaccine development has been so difficult for so many years, and we'll see why that is in a minute. And coronavirus, hopefully they will get a vaccine pretty soon. The chances are much better to get an effective vaccine for that disease. So anyway, so let's take a look at preventing and treating HIV/AIDS. Of course, safe sex, that's been sort of the slogan for many years, using condoms and also discouraging sexual promiscuity. That's all good advice. Using sterile needles every time. Do not ever share needles. That for IV drug users, of course, you have to use a sterile needle every time. Stuff gets transmitted, other diseases besides HIV/AIDS, with needle pricks, of course. And then the availability of medications gotten better in recent years. So when Magic Johnson first announced that he was HIV positive, they gave him 10 years. Well, he's still alive, doing well because of the new medications. So there are now good medications available, especially the cocktail that we'll just go over in a minute. HIV/AIDS is now considered a treatable chronic disease in the developed world due to the availability of medications. There's a variety of medications available, such as fusion or cell entry inhibitors. They target a GP1 anchor protein, and that prevents infusion of the virus with the cell. And so here are some brand names of these medications. And then here's a variety of other medications. Reverse transcriptase inhibitors, that was one of the early ideas, and definitely a good idea since it's a retrovirus and it relies on reverse transcriptase to convert its viral RNA back into DNA for integration into the host cell genome. And so if you have a reverse transcriptase inhibitor, that's the way to go. So you have here these nucleoside reverse transcriptase inhibitors and RTIs. Here are some brand names for that. And then non-nucleoside reverse transcriptase inhibitors. Here are some brand names for that. Really, none of these individual medications have proven super effective. Out of an act of desperation, there was somebody that said, okay, I'm just going to put everything on the shelf that I have in antiretroviral medicines. I'm going to put everything into one cocktail and just see what happens. And that ended up being this highly active retroviral therapy or HAART therapy. So basically, in an act of desperation, somebody threw everything that they had available in antiretroviral medicines into one cocktail and gave it to patients, and that ended up being the ticket. So you're using a combination of drugs to minimize survival of resistant strains. And here are some more treatment options, and they're all part of this highly antiretroviral cocktail. Integrase inhibitors, they inhibit HIV integrase that integrates the cDNA from the RNA, the viral RNA, into the host cell genome. Here's the brand name for this. Protease inhibitors, a great idea because that inhibits the proteases that cut the protein coats for the virus. Here are the brand names for that. And then maturation inhibitors, tetherins, they tether viruses to the cells, preventing the release and the spread of the virus. And you put all of that in one cocktail, and there you got your HAART therapy. So here are the drugs that inhibit the HIV life cycle. You have fusion entry inhibitors, and here are the brand names for that. So you want to prevent fusion or entry of the virus into the host cell. Then the reverse transcriptase inhibitors, it's very logical because the HIV has RNA as its genome, and you need to reverse transcribe RNA into DNA before it can be integrated into the host cell genome. So if you inhibit that enzyme, the reverse transcriptase inhibitors, it completely makes sense. Then you have integrase inhibitors, brand name right here, and that's a good idea too. Then the protease inhibitors, they prevent the cutting of the correct protein coats, and they prevent then this assembly of the virus into functioning viral particles. And there are some of the brand names listed right there. Now, of course, we know that your best bet against a viral disease is vaccination. Antibodies, if you have them before the virus hits, and that's the ticket, because then the virus can't establish itself in your body, and you prevent the disease. But developing an HIV vaccine has been elusive. It's been very challenging, and that's because this virus changes a lot. It's a retrovirus, reverse transcriptase. That enzyme makes a lot of mistakes. That leads to mutations. This thing changes all the time. You also have no model of natural immunity that you could use as sort of a guideline. You don't really have animal research available. There's still a lack of understanding of some of the mechanisms of retroviruses, then this high mutation rate, then resistance strains. That is really a problem. You may develop a vaccine, but the next thing you know, the virus is mutated and the vaccine is no longer effective. An ideal vaccine would induce immunity before reservoirs of latent virus are established, stimulate production of cytotoxic T lymphocytes, and of course needs to be affordable. So far, no success. Hopefully, we're going to be doing better with the coronavirus vaccine that is in development, hopefully right now. But anyway, this concludes our discussion. Whoa, this was my cat. So this concludes our discussion of the lecture portion of microbiology, and I hope you enjoyed our journey here online. So I have a few more words to say about... Oh, my cat is going crazy. Sorry about that. So the cat calmed down right now. But anyway, what I wanted to tell you is how to prepare for the final exam. You have a 200-point final exam coming up, and it will cover the entire course. That means it is comprehensive. So what you should do is review your notes for the previous 3 midterms that have covered most of the material minus the very last lecture. But anyway, the final exam, it is all multiple choice questions, matching sets. There is no fill in the blank. There's no written portion to this final exam. It's just 200 Canvas graded, typically otherwise would be Scantron graded questions. Now, you do have your notes available, and so just make sure that you're organized. There are some pathogen questions, quite a few actually, on the final exam. Make sure that you do the pathogen test should be first. Make sure you have that information handy also for the final exam. The way to prepare for the final exam would be to review each chapter that we covered, and I would go over the lecture notes that you took first. Then I would read in your Tortora textbook, read the end of chapter summaries and do the multiple choice questions that are at the end of the chapter. Those are really good preparation. You have the answers available for those questions. They're part of the textbook. All you have to do is look it up. And so use these questions as preparation questions. And pretty much have your pathogen charts ready, handy to look up. Take notes, whatever you think you want to have handy right away, stuff that you can't remember. Writing notes is a great way to learn material. Make sure that you are very organized, of course. So review each chapter, review your old notes, review the lecture material, the end of chapter summaries, and the questions that are going along with that. and you should be fine. I'm pretty sure you'll be doing just fine. For the most part, you guys have been doing absolutely awesome. I am very proud of you that you were stuck, that we stuck it out, and we finished this class online. So taking microbiology online was not easy for sure, and you guys were right here with me the whole all of the way, and so we got it done. You did get some solid lab hours in the lab in the beginning, and this was really unfortunate that we had to take microbiology online, but I think the lecture portion was okay. The lab, I hope you enjoyed the labster portion of this and got something out of that. I did the best I could. Now it's your turn. Now you do the best you can, and I'm sure you can finish strong and get this final exam over with and get the grade that you want and that you deserve. Okay, so hang in there. You can still text message me or email me if you have any questions. Log in for the office hours that are available, and just work hard and get it done, and you will move on and go off to bigger and better things. All right, take care now.